Hello students, in our today's lecture we will discuss about the cell wall, that is outermost boundary, not in all cells but in especially in plant cell, in fungal cells and in prokaryotic cells. So in animal cells it does not present, and there are three types and it is secreted by the protoplasm. What is protoplasm actually? uh, what is protoplasm is actually the word protoplasm and protoplast are different, and you know that cytoplasm plus nucleus is equal to the protoplasm. So you can say cytoplasm plus nucleus is protoplasm, or living matter of the cell is protoplasm, and protoplasm plus cell membrane is actually the protoplast, or we can say that plant cell with the cell wall removed is called the protoplast. So the next is the types of cell wall. These are three types, which are the primary wall, secondary wall and the middle lamella. From going to discussion, we will first see the pictures of these three types of the cell wall. So here we have the picture that there are three cell of the plant cell, and you can see the outermost boundary, that is the primary cell wall, and inside it you can see it is the secondary cell wall, and you can see it is a dark red, that is the secondary cell wall, and the inner yellow color is actually the cell membrane or plasma membrane, and inside is the cytoplasm. And in between the cell you can see this yellow or dark yellow color. This is actually the middle lamella, or you can say it is a middle membrane that is present between two plant cells. So here we can see the primary cell wall. The primary wall is actually called true wall, and it develops in the newly growing cells, and it consists of three major things: cellulose, pectin and hemicellulose. And the cellulose that is present is in the crisscross manner. Crisscross means they have the fibers that are perpendicular to each other and having angle of 90 degree. So in the primary cell wall, cellulose, pectin and hemicellulose present, and cellulose fibers are in present crisscross manner. And the middle lamella is between the cells, and the third one is the secondary cell wall that is formed on to the its inner side. Actually, as we can see in diagram in details, that the secondary cell wall is present inside the primary cell wall. So it is comparatively thick and rigid, and composed of many inorganic salts, silica, vaccine and cutin, lignin, etc. So you can see the composition of primary and secondary cell wall are different. So let's start with the reading. The outermost boundary in most of plant cell is cell wall. Cell wall of the plant cell is different from that of prokaryotes, both in structure and chemical composition. It is secreted by protoplasm of the cell. Its thickness varies in different cells of the plant. It is composed of three main layers: primary cell wall, secondary and middle lamella. Middle lamella is first to be formed between the primary walls of the neighboring cells. Primary wall is composed of cellulose, pectin and hemicellulose. Cellulose molecules are arranged in a crisscross manner. The primary cell wall is a true wall and develops in the newly growing cells. The secondary cell wall is formed on its inner side and comparatively thick and rigid. Chemically, it is composed of inorganic salts, silica, vaccine, cutin, lignin. Prokaryotic cell wall lacks cellulose. Prokaryotes does not contain the cellulose. Actually, they are composed of some sort of the polysaccharide subunits. These are called the peptidoglycans or murin, and the fungi contain chitin instead of cellulose. Prokaryotic cell wall lacks the cellulose. Its strengthening material is peptidoglycan, and the fungal cell wall contain chitin. And we can also mention here one thing more, that the cell wall is actually non-permeable. It does not pass different materials to pass through it, does not allow. So here we can see cell wall is very important. It provides the definite shape to the cell wall and keeps it rigid. It does not act as a barrier to the material passing through it. Sorry, I have previously said that it does not allow substances. Actually, it is a freely permeable. Sorry for that. Freely permeable means it allows all substances to pass through it. The next is the cytoplasm, that is very important with the MCQ point of view and short question. Cytoplasm actually is the material that present around the nucleus and inside the cell membrane. As we have discussed in the cell theory, the three components of the cell membrane, a cytoplasm and the nucleus, actually the liquid material that present inside the whole cell, it is called the cytoplasm. And when we add the nucleus and cytoplasm, it is called the protoplasm. And it consists of many soluble substances and many insoluble substances. So the ground substance is formed by the soluble substances and it is called the cytosol. And there are few substances which form a true solution, and there are few substances which form the colloidal solution. Actually smaller substances form the true solution and the larger substances form the colloidal solution. So the colloidal solution are of two types. It may be viscous or it may be non-viscous. Actually it may be sol or gel. If it is a non-viscous, it is called the sol. You can remember it by saying the solution. And it may be the viscous. So peripheral parts are normally gel. And what is the function? Actually it provides the site of the different function, and it provides the site of the several type of the vital chemicals, and it stores a lot of things. So let's start with the reading of cytoplasm. The living content of eukaryotic cell are divided into nucleus and cytoplasm. The two collectively form protoplasm. Cytoplasm consists of an aqueous ground substance containing a variety of cell organelles and other inclusions such as the insoluble waste and storage products. The soluble part of the cytoplasm is called cytosol. It forms the ground substance of the cytoplasm. Chemically, it is 90% water. It forms a solution containing all the fundamental molecules of life. In the cytosol, small molecule and ions may form true solution, and some large molecule form colloidal solution. Colloidal solution may be sol, non-viscous, or it may be gel, and peripheral parts of the cell are like gel. The most important function of the cytoplasm is to act as a storehouse of the vital chemicals. It also a site for certain metabolic process such as the glycolysis. So cytoplasm contain a lot of organelles as well. There are a lot of organelles like mitochondria and all the organelles of the cell: Golgi apparatus, nucleus, plastids, ribosome, lysosome, centrioles. So but they are not all the organelles are freely moving. There are some organelles that are moving inside the liquid of the cell. These are especially mitochondria that move in the cell. In living cell of the cytoplasm contains several cell organelles such as endoplasmic reticulum, mitochondria, Golgi complex, nucleus, plastid, ribosome, lysosome, and centriole. The free floating cell organelles, for example, mitochondria, move about in the cytoplasm due to cytoplasmic streaming movement, and this is the active mass movement of the cytoplasm. In the next lecture, in the next our top part of the lecture, we will discuss about the endoplasmic reticulum. Actually, what is the endoplasmic reticulum? Actually, it is nothing but a network of tubules that are spreading from the nucleus to the cell membrane. So these are channels, actually these are tubes actually, that extend in the whole cell, you can say from nucleus to the cell membrane. And they have the some sac like structures that separates the material inside it from the cytoplasm. The material that is present inside is separated from the cytoplasm. As it is a tube, it has some material, and it is separated from the cytoplasmic material with the help of units called the cisternae or the tubes. So there are two types of the endoplasmic reticulums. Actually there are the smooth endoplasmic reticulum and rough endoplasmic reticulum. So as the name represents smooth, it has smooth appearance and does not contain ribosomes on the structure. But the rough endoplasmic, as the name represents, the rough means actually the ribosomes are attached onto their structures. And as the name represents, as the ribosomes are attached to it, so these are the rough endoplasmic reticulums. And they are involved in the protein synthesis. And the smooth endoplasmic reticulum does many functions. For example, the lipid metabolism or the breakdown of lipids is acted upon by the smooth endoplasmic reticulum, and many toxic substances may enter into the cell, and these detoxify actually them. And two more functions, that are the transmission of nerve impulse, or you can say the message. The message is transferred from one part of the cell to the another part of cell through the smooth endoplasmic reticulum, and transport of many materials, you can say entering by entering into the one side and then the exit from the other side. So these are the four functions: lipid metabolism, detoxification, transmission of impulses, and transport of material is done by the smooth endoplasmic reticulum. So let's start with the reading. Under an electron microscope, a network of channels is seen extending throughout the cytoplasm. These channels are often continuous with the plasma membrane and also appear to be in contact with the nuclear membrane. This entire system of channel is the endoplasmic reticulum. These membranes vary widely in appearance from cell to cell. The material present in these channels is separated from cytoplasmic material by the spherical or tubular membrane. These are called actually cisternae. You should not confuse it with the cristae that is present in mitochondria, but here we have the cisternae. There are two morphological forms of endoplasmic reticulum: a rough form with attached ribosome and smooth form without ribosomes. The rough surface endoplasmic reticulum is involved in the synthesis of protein. After synthesis of protein, they may be either stored in the cytoplasm or exported outside the cell through these channels. The smooth surface endoplasmic reticulum help in metabolism of the number of different types of molecule, particularly lipids. They also help to detoxify the harmful drugs in some cell. Smooth endoplasmic reticulum is responsible for transmission of impulses, for example, in muscle cell or nerve cell. In addition, SER also plays an important role in the transport of material from one part of the cell to another part. Endoplasmic reticulum actually maintain the cell shape, and the cell wall actually gives it shape. So the difference should be clear. The cell wall actually provide the shape to the cell, and endoplasmic reticulum support the cell shape, or we can say shape maintain. Thank you, take care, study well, and for further these types of videos in which you should have a clear concept regarding your biology. So thanks for watching, and subscribe my YouTube channel and press the icon for my new videos that may be effective for you. Thank you.